Here are the answers to the critical thinking practice for Chapter 12. For the first question, I ask what the pHs of the granules in the various granulophiles are. Eosinophiles, or sometimes called acidophiles, basophiles, and neutrophiles. So this may seem like an impossible question, but what it really does is ask you to think back to the chapter on chemistry when we talked about ionic interactions. Acids have what charge? That's right, they have a negative charge. And bases have what kind of charge? Positive charge. And those negative and positive charges are attracted to each other. So, also going to the chapter when we talked about the Gram stain and the structure of cell walls, the outside of most cells have what charge? Negative. So what charged dye do we use to stain the actual cell? We use a basic stain because it's attracted to the negative stain on the cell, stains the actual cell. Then if we do a negative stain and we want to stain everything but the cell, we use a stain that has a negative charge because the two negatives are repelled. Alright, now back to the actual question. So these are named by how they react to various stains. Eosinophils stain with eosin, which is an acidic stain. Basophils stain with basic stains. And neutrophils stain with a combination of both. So, does that help you answer the question? The answer is eosinophils have basic granules because they stain with an acidic stain. Basophils have acidic granules because they stain with a basic stain. See, the opposing charges, the negative charge of the negative stain, the eosinophil, is attracted to the positive charge of the basic granules. The basic stain with the positive charge is attracted to the negative stain, the negative charge of the granules, and neutrophils, of course, are neutral. So, how do we know? Well, because of the discussion that we just had on the previous slide, it's because of how they respond to various stains, partially because of the research that's been performed on these types of cells. Part of the research is how they respond to the various stains, and partially because of how they're named. These two go together because of this. Now for this question, how would the pH affect the functioning of the granulocytes? Once again, I want you to go back to that chapter on chemistry, and also when we talked about being able to grow microbes. It's all about pH. So, acidic and basic molecules can denature and otherwise disrupt molecules of pathogens. True. Basic molecules stimulate the innate immune system better than acidic molecules. There's no evidence for that. Acidic molecules stimulate the adaptive immune system better than basic molecules. There's no evidence for that either. But the enzymes inside the granules function best at those pHs. That is a true statement. Alright, enough with pH. Now, onto this question. What would be the effect of a patient if they had a mutation that led to reduced numbers of natural killer cells? For this question, you need to think backwards. You need to think about what natural killer cells do and how that would affect the patient. If we took out natural killer cells, how would we tell? First, what do natural killer cells do? Natural killer cells attack any cell that's displaying pathogenic markers, in other words, saying, "Help, I'm infected," or cells that aren't showing enough MHC marker. In other words, enough markers that say, "Hey, I'm a part of the human body, I should be here." Well, generally speaking, viruses are the pathogens that get inside of cells, infect cells, and cells display on their outsides viral markers, viral antigens. Also, cancer, part of what makes a cancerous cell cancerous is they don't make molecules that they should be making because they're too busy dividing uncontrollably. And one of the molecules that they don't make are MHC molecules. Cancer cells have a reduced number of MHC on their surfaces. So, if we didn't have natural killer cells, what would we have? Well, we'd have an increase of viral infections because the natural killer cells wouldn't be killing those cells that were displaying viral proteins on their surfaces. And there would be an increased occurrence of tumors because the natural killer cells are not getting rid of cells that don't display enough MHC. In other words, they're not getting rid of cancer. Now, for this next question, I asked a variation on this. What would happen if you had a reduced number of neutrophils? Once again, you got to think backwards, starting with what do neutrophils do? Well, neutrophils phagocytize. They are first responders to the site of inflammation. There have been cytokines released because there have been pathogenic molecular markers, or PAMPs, noticed, and all sorts of other stuff. Also, if things get really bad, then they will explode in a net and throw their DNA that's been mixed up with the granule contents, the poisonous, toxic granule contents, over the pathogens. So, the question is, if we didn't have these first responders, what would we have? Well, we would probably have increased bacterial infections because bacteria, most of them replicate outside of the cells, but they do have pathogenic markers, they do have PAMPs, and that will stimulate inflammation. Probably wouldn't show an increase in viral infections because natural killer cells take care of those. So, increased bacterial infections. Maybe increased fungal infections, but they do display pathogenic markers, or PAMPs, and that's probably what we would have. Now, I do warn you that I like this question, and you may see either this question or the previous question on an exam, or the question asked a different way, so that one of these two are the correct answer.